Good morning friends. Today we will learn about the another topic of genetic engineering, that is cDNA library, which is the part of a Unit 1 course RBT 501. I am Dr. Deepak Gola, Assistant Professor of in the Department of Biotechnology, Noida Institute of Engineering and Technology. So, first of all, what is cDNA library? cDNA library is like a collection of fragments that have been cloned into the vector that have been cloned into the vector, so that researcher can identify and isolate the DNA fragment that interests them for the future studies. These are basically are of two kind of library. First is the DNA library, genomic DNA library, and second is the cDNA library. So, genomic DNA library contain both the whole gene with the exon and intron, where basically the cDNA library contain only the coding part from the DNA. So, so how will first of all we have to isolate the RNA for making the cDNA, So, for the synthesis of complementary DNA, we have to isolate the messenger RNA template, and we have to utilize some enzymes to make the cDNA from that. So, these are the steps for the construction of cDNA library. First of all, the first point is the isolation of messenger RNA. You have learned about the messenger RNA. It is involved in the formation in the synthesis of the protein with the help of tRNA. Messenger RNA will code the protein, different kind of protein which are utilized in the various biochemical processes. The second is the synthesis of the strand. As we know, the messenger RNA is single stranded, but what we require DNA, we know that DNA is double stranded, and to fix that into the vector we require to make it a double stranded. The next step is the incorporation of that double stranded cDNA into the vector, and vector, in the previous slides we learned that vectors are the like the vehicle which are used to carry the desired DNA, a part of DNA into the bacteria or into the back, yak. There are different type of first of all we have to after the synthesis of strand, we will incorporate DNA into the vector. Vectors are known as a vehicle which carry the DNA into the into the bacteria or fungi. The next is the cloning of the cDNAs, that is complementary DNAs. So, first of all we have to isolate the messenger RNA, and for that we require a chromatographic purification. Here what we have to do, these are involve some steps. So, it involves the isolation of total messenger RNA from a cell type or a tissue of interest. The amount of desired messenger RNA can be increased by the following steps. First of all, the chromatographic purification of messenger RNA utilizing the oligo T, which retain messenger RNA molecule, resulting in the enrichment of the aliquot of the messenger RNA. Second is spinning down the messenger RNA by density gradient centrifugation. Next is messenger RNA preparation from a specialized cell like developing seed, chicken, oviduct, and erythrocyte B cell of pancreas, because these are rich in protein, and what make protein is the messenger RNA. So, the next step is the ends of eukaryotic messenger RNA consist a string of 50 to 250 adenylate residue, which is also called poly A tail, which make the separation easy from the much more prevalent rRNA and tRNA in the cell extract. As we know, when we isolate, it will contain different kind of RNAs like messenger RNA, tRNA, rRNA. These are all type of RNA which are present in the cells, but we want only messenger RNA. So what we will do, we will utilize the poly A tail of this messenger RNA to isolate the messenger RNA from different type of RNA. The bound messenger RNA can then be eluted using the low salt buffer. So we will discuss the step in detail here. So this is the shape of tRNA, this is rRNA, and this is messenger RNA. So here we know at the 3' end, this is, if this is a messenger RNA, it should have 2 ends, that is 5' end and the 3' end. So the 3' end contain a tail of adenine residue. So we know there are different type of purines and pyrimidines, and adenine is one of them. So we will utilize this part to isolate the messenger RNA from the solution of different RNAs. So what we will do, we will use a oligo T matrix. This is the oligo T matrix. It is like a structure on which we have immobilized our primers, that is basically containing only T, that is thymine, the like 50 to 100 base pair of thymine. So when we will pass through, when we will let this be a column and this is the matrix with containing TTT, and this is the messenger RNA and it contain adenine. So what it will do, that adenine of poly A tail will bind with this T, and it will bound to the matrix, and rest of that is tRNA and that is rRNA. It will move out from the elute out from the solution. So as we can see here, this adenine is bound to TT in the hybridization condition, like specific temperature or pH or buffer it is required. So it will bind with this T, the adenine, and rest of the RNA like tRNA and rRNA will wash away. So after this bound, we can apply like low salt buffer. So what low salt buffer will do, it will break this low salt buffer will break this bond, and when this bond is break, so automatically the messenger RNA will elute out messenger RNA from the matrix. So it will elute out the messenger RNA from the matrix. So this is the first step, that is the isolation of the messenger RNA from the solution of DNA RNA. Then the next step is the synthesis of the strands. As we know, complementary DNA will require is the double stranded and RNA is the single stranded. So again we will use we will make use of the poly A tail. So it lie in the 3' end, and we can see this is the oligo T primer. So it will bind with the adenine part, that this T, and we will utilize the viral reverse transcriptase. So what this viral reverse transcriptase will do, it will synthesize the second strand from this 5' end to in this direction to 3' end. So it will synthesize, this is 5' end and this is 3' end. So it will synthesize that that required strand from in the direction of 5' to 3' end with the help of viral reverse transcriptase. So, let it be this blue color is the strand which is synthesized, but what viral reverse transcriptase do, when that there is no leftover residue at the 5' end, it will loop back. So, it will form a hairpin loop at 3' end. So, now as we know RNA what RNA contain? RNA contain uracil instead of thymine, but DNA contain thymine. So, we have to remove this uracil also from the strand. So, what we will do, from the this red color strand, it contain uracil like this, like in the strand. So, we will degrade this strand and we will form the new strand utilizing that 3' end. So, what we will do, first of all we will degrade this RNA strand with the help of NOH. This is a form a alkaline solution and it will degrade that RNA. The next step will be utilizing a DNA polymerase. You have heard about this enzyme in the replication part. So, what will do, it will utilize this 3' end and it will, what it will do, it will synthesize the new strand in this direction. So, it will form a like this hairpin loop like structure. So, this hairpin loop like structure is our cDNA. So, what will the next step will it contain, like this is a loop. So, we have to degrade this loop. So, we will utilize another enzyme in this process, that is S1 nuclease. What S1 nuclease will do, it will degrade, it will cut down this part, it will act only on the loop part and it will degrade, and what we will get is a double stranded DNA, or in this case we will call this as a cDNA. In this case we will call this as a cDNA. So, the next step is incorporation of the cDNA into the vector, but as we can see there is no for inserting into the vector, we will require what? We will require the some like ends, that is called blunt end. We will require some blunt end. So, for this creating a blunt end in the cDNA, we will require another like part, that is called linker. And what is linker? It is a double strand DNA segment with a recognition site for a particular restriction enzyme. Sorry. So, it is a 10 to 12 base pair long prepared by hybridizing chemically synthesized complementary oligonucleotide. The blunt ended double stranded DNA are ligated with the linker by with the help of, with the linkers by DNA ligase enzyme from T4 bacteriophage. So, here is the example how we will ligate. Let it be this is the linker, let it be this is the linker, and it contain a restriction site for like restriction enzyme for a site for a restriction enzyme, and it will cut down like from here and here. So, we have to ligate this to the ends with the to the ends of complementary DNA. So, let it be this is the cDNA. So, what will we do? We will ligate these oligonucleotide which contain the restriction site, also called linkers, to this part with the help of T4 ligase. So, then after that we will after addition of these linkers, we will digest this with the particular enzyme. In this case we are taking E. coli because it this linker contain the restriction site for eco R1. So what we will do after like applying the enzyme eco R1, it will cut down the DNA and will create the cut down the DNA from restriction site and will create the ends, that is called sticky ends. These are called sticky end, and it will be helpful to have a sticky ends because it can ligate easily now into the desired vector. So there is one more problem. If our like this is our cDNA with ends. So what if cDNA also contain a sequence for eco R1? Then what we will do? So because if we apply the eco R1, it will cut down the complementary DNA and it will be of no use because we require the whole complementary DNA. So what we will do? We will utilize another enzyme which is called eco R1 Methylase. So what eco R1 Methylase will do? It will methylate the complementary DNA at the adenine site, like we can see it is methylated. So if it contain the restriction site for eco R1 and it get methylated, restriction enzyme will not cut the methylated part. Restriction enzyme will not cut the methylated part, it will only cut the non-methylated part. So that is how we will avoid the cutting of our complementary DNA via eco R1 during the cDNA library preparation. So after the cDNA library is prepared, here we have our messenger RNA, and with the help of reverse transcriptase we prepared our complementary DNA. So digestion with the restriction enzyme will cause like it will produce the fragment, and these fragment what we will do, we will combine these fragment to the vector and ligate them into the vector and it form the recombinant vector. Now our complementary DNA is present here. So we have to transform this recombinant DNA into the desired bacteria. So it will produce its own clone with the help of bacterial machinery. So this is the basic step. It is same as that of cloning of a gene, but in this what we do, we have to prepare the complementary DNA from the messenger RNA. So this is the extra step, otherwise it is same as that of genomic DNA library. So the preparation of complementary DNA with the help of different enzymes is there in this process. So what is the problem with cDNA preparation? So there are a lot of problem when we create a cDNA library. So first of all, the large messenger RNA sequence result in ineffective synthesis of full length of complementary DNA. So this cause the problem during expression, as it may not contain the entire coding sequence of the gene. So as we know, gene will code a particular messenger RNA, from that we will get the protein. So sometimes the sequence of the gene is too large and we are unable to synthesize the full length of that full length complementary DNA from that messenger RNA due to its long size. So we will during the preparation of cDNA library, we will not get the entire sequence of that gene. Second is the use of S1 nuclease. For this to understand, we will go back. So what this S1 nuclease will do, we can see here that this part is the part of RNA, and when we apply the S1 nuclease, it will cut down this some base pair from this side. So it will be the it will like it will also remove some base pair from the ends. So the enzyme used to trim the DNA double stranded complementary may remove some important base pair from the 5' end, which is required to for some analysis, like for the functioning of the gene or functioning of that protein. So, and in the next step we will learn about the application of cDNA library cloning, cDNA library cloning. First of all, it help in the discovery of novel gene. Second, it help in vitro study of gene function by cloning full length complementary DNA. Next is the determination of alternating splicing in a various cell type or tissue. Then they are commonly used for removal of various non-coding region from the library. As we know, non-coding region, we know genomic library will contain both coding plus non-coding, but cDNA library will contain only coding part. Then the next is the gene expression required either for the detection of a clone or the polypeptide product may be as a primary objective of the cloning. Next is to study the expression of messenger RNA. Thank you, and for more detail you can contact me on the given email ID.